Hi, my name is Zhe Wang. I'm a research fellow from the National Institute of Biomedical Imaging and Bioengineering at the NIH.
Hi, my name is Xiang Qian. I'm a senior investigator and the chief of the Laboratory of Molecular Imaging and Nanomedicine, National Institutes of Health. Today, we would like to share with you our recent work published in ACS Nano using polymeric nanovehicles to regulate and track the differentiation of transplanted neuron stem cells in a mouse model of traumatic brain injury, or TBI.
Stem cell therapies have drawn much attention due to their multifunctional ability to assist the recovery from diseases and injury, including those affecting the brain. One challenge of applying neuron stem cells in TBI recovery is to direct cells to differentiate into appropriate neuronal subtypes, with the objective of compensating for lost brain function.
The second challenge is to develop a non-invasive imaging method to monitor the differentiation of neural stem cells after transplantation into the brain.
To facilitate neuron stem cell differentiation, we have developed a biodegradable polymeric nanovehicle that encapsulates a water-insoluble morphogen, retinoic acid, and provides sustained release of that molecule. We have shown that this nanovehicle was efficiently taken up by stem cells. This provides a sustained concentration of retinoic acid in the neuron stem cells and increases the propensity for differentiation. In TBI mouse model, we administered neuron stem cells that had been pretreated with this nanovehicle and show the differentiation of transplanted cells into neuron and astrocytes. We also show that cells stimulated by retinoic acid through this nanovehicle have accelerated recovery of synapse-related proteins and rescue of gene expression that cooperatively ameliorate the impacts of brain injury.
In order to address the second challenge, we have created a gene vector that we call TUPIS, T-U-P-I-S, which encodes a reporter gene whose expression is under the control of a neuron-specific promoter. The reporter gene codes for a unique enzyme that can be imaged using bioluminescence and thus allows non-invasive imaging of the differentiation of transplanted neural stem cells. Using bioluminescence imaging, we observed that neural stem cells undergo migration, proliferation, differentiation, and apoptosis after transplantation. A handful of stem cells ultimately take up residence in the injured area and evolve into mature neurons.
In this study, we have implemented two new techniques. The first, a polymeric nanovehicle technique, can provide a broadly adaptable nanoplatform that can sequester various water-insoluble bioactive molecules, including the morphogen, retinoic acid. The nanovehicle provides a sustained morphogen release pattern in order to ensure continuous, steady, in vivo stimulation of cells that can be differentiated into appropriate neuronal subtypes. The second technique, our neuron-specific reporter gene coding vector, can be used to derive reporter genes for bioluminescence, fluorescence, and radionuclide imaging.
Although stem cell transplantation for brain disorder therapy is at the frontier of regenerative medicine, our reported work provides a new understanding of the fate of neural stem cells after transplantation. Many unknowns remain to be explored in order to achieve the full potential of nanomedicine in stem cell therapy. Our study provides a prototypical example of nanomedicine in neuroscience. It is our hope that the chosen nanomaterials and the morphogen release patterns from our nanovehicles can provide guidance on designing additional nanomaterials for stem cell labeling and stem cell therapy.